In this chapter, we're going to cover the central nervous system. We're going to get an overview of your central nervous system, which consists of two parts, your brain and your spinal cord. In this first portion, we'll really cover the anatomy of the brain. We'll discuss how the brain is protected, and we'll look a little bit at the anatomy in lab. Then for the second portion, we'll cover your sensation and movement, how the brain is responsible for maintaining homeostasis, cover a little bit of the higher mental functions, and then end with a look at your spinal cord. So the central nervous system overall does contain the brain and the spinal cord. It's involved with not only movement, but helping you understand information coming into your body, your sensory input, maintaining homeostasis, that balance of things, and functions relating to mind like thinking and processing and speaking, emotion. That's all the job of your central nervous system. When we look at the central nervous system, the brain and the spinal cord, we'll see that there's two types of matter in it. Now, it's mostly made of neurons still, but those neurons can come in two forms. You can have neurons with myelinated axons, that fat-filled wrapping, and that myelinated axon gives you what we call white matter. This was a review from a previous chapter. In the spinal cord, your white matter is all superficial. It's on the outside. This is the spinal cord here, so you have white matter and white matter and white matter and white matter, and that's it. But in the brain, the white matter is deeper because you first have a section of gray, and then you have gray again. So in the spinal cord, our white matter is superficial, whereas in the brain, we refer to the white matter as being deep. Gray matter would be the neuron cell bodies, the dendrites, and the axons that don't contain myelin. In the brain, you have gray matter, as I just said, in two spots. We have it here in the outer cortex, and we have it here in the inner nuclei. So the cortex here, and on the inside. In the spinal cord, you'll see the gray matter is also very deep. And in this case, it's shaped kind of like a butterfly, you'll see here. In the brain, the gray matter is going to be scattered, whereas you see it much more concentrated. In the spinal cord, that's where the gray matter is going to be processing information. It's surrounded by what we call tracts, or bundles of white matter on the outside, so they can relay information. We want to focus then mostly on the brain. This soft, whitish, grayish organ that's going to connect with your spinal cord, live within your cranial cavity, and directly or even indirectly control every single one of our body functions. Your brain itself is not very heavy. Here's a gram reading, but for those not comfortable with the metric system, it's about 3.5 pounds, mostly made of nerve tissue, but there is some epithelial, there is some connective tissue to help support the brain. The brain is also not one solid organ. It has cavities on the inside referred to as ventricles, and these ventricles are filled with your cerebral spinal fluid, which is going to behave like shower water for our brain. It's going to help clean the brain and do several other functions for it. Most of the brain is going to need huge amounts of oxygen and huge amounts of glucose at all times. It's a really big ATP organ. About 20% of its total blood flow, though, is going to come when it's at rest, but we really need a lot of blood flow here. Four main parts to your brain. We have the cerebrum, which is the outermost part that's very visible. The diencephalon, which is internal, so that's kind of shaded because it's inside. The cerebellum, which is here in the back, and then the brain stem here at the bottom. So four major regions: the very large in volume cerebrum, the smaller cerebellum, although it does have about half of the neurons, and the spinal cord, the brain stem connecting to the spinal cord, and then the smaller internal diencephalon. We'll start with the cerebrum. This is what's responsible for your thinking, your interpretation of stimulus coming in, your planning what you're going to say, planning what you want to do, you're initiating the cerebellum for you to move. If I look at the anatomy of the cerebrum, it's a very folded organ. So remember why we would ever fold anything in the body. we want to increase the surface area. In the brain, we have these folds called a gyrus, which creates these little grooves called a sulcus. So I'm going to take this and pull it out some. If this is the gyrus, this would be the gyrus. That would mean this would be the sulcus. Although it's not wide, it's actually going to sit like this in the brain. So the gyrus and the sulcus, and the gyrus and the sulcus. The sulci would be the shallow grooves. We will put blood vessels within those. They're going to stay very well protected running in the sulcus. The gyrus is going to help you increase your surface area because we really do have a limited amount of space inside the cranial cavity. We also see throughout the cerebrum that we have some fissures, some very deep grooves. The most notable one runs right down the middle, and it's called the longitudinal fissure. Now, if I were to stick my fingers down into your longitudinal fissure, I could take your left hemisphere and your right hemisphere and pull them apart, but not completely. You can pull the right and left cerebral hemispheres apart up to the connection between them, referred to as the corpus callosum. But it is a very deep groove that does break the left and the right hemispheres. Inside of the cerebrum is where I would find my ventricles, these cavities internally that contain your cerebral spinal fluid. The 5 lobes can be distinguished from each other by using our sulci, those grooves, the gyri, the folds, or the fissures, the very deep grooves, as kind of landmarks. If you look in the brain, you can see that there is a central sulcus that runs almost straight. That will help you separate your frontal lobe from your parietal lobes. Then I have a fissure, a deeper one here on the side, that can help me separate the temporal from the previous 2. And then if I were to take forceps and pull it back at that fissure, I would see the insula buried deep within. On the posterior surface of the brain is the occipital lobe. Now, in the brain, we don't really refer to them as anterior and posterior. We refer to these directions as rostral and caudal. I'm sorry, my pen will not cooperate and write that A. Clear it off. I'll try again. We have the rostral and we have the caudal. Caudal means tail, and if you think about rostral is over here on the side where your nostrils are, that's the front. So the occipital lobe is the caudal portion of the brain. The frontal lobe is the rostral portion of the brain. The parietal lobe sits superior. Your temporal lobes are lateral, and your insula is also buried deep within on the lateral surface there. Each of these lobes have a job. The frontal lobes are responsible for your planning. Okay, class starts at 8 a.m. I live 20 minutes from campus. I better leave home by 7:30. Gives me 20 minutes to drive, 10 minutes to find a parking spot. That's the job of your frontal lobe. Executing movement, which would communicate to your cerebellum, is also the job of the frontal lobe. Your behavior, your mood, your personality, your conscience. Is this right or wrong? That's all the job of the frontal lobe. There's a lot of psychology studies about people who have a damaged frontal lobe and how it alters their personality. Phineas Gage is a very common example used of how damage to his frontal lobe completely changed his personality. The parietal lobes, as I said earlier, are just behind the frontal lobes and they're sitting superior. These are responsible for processing and integrating input information, sensory. They're also what helps us pay attention. Their function in attention span. Your temporal lobes on the lateral sides are, of course, hearing. Your ears are there. They're also your language centers, where we plan what we want to say. We make the motor action to say what we want to say. Memory is here in the temporal lobe, as well as a part of your emotional content here in your temporal lobe. Very interesting studies about people in car wrecks and how hitting their head on the side of the window created memory deficiencies, created amnesias, and things of that nature. It's one reason why we now have side curtain airbags. Your occipital lobe back there in that caudal posterior region of the brain is all for vision. The optic nerve coming out of your eye goes straight to that occipital lobe so we can process visual input. Now those lateral fissures, those deep grooves, if I pull them apart, I can find the insula. Now the insula is one of the newest parts of the brain that we've begun to study. Only in the last couple of years have we even understood what the insula is potentially doing. We believe it's relating to taste and to the control of your viscera, your internal organs in your thoracic and abdominal region. The cerebrum also has this section referred to as the limbic system. Now it's a system, so it's made up of different parts. It's the gray matter in the limbic area, along with a structure referred to as the hippocampus and the amygdala and the pathways that connect them. So here's the hippocampus, here's the amygdala, and this is what we refer to as the limbic lobe. These are regions within the cerebrum. You can see they're in the frontal, as well as the parietal, as well as into the temporal lobe. The limbic system is a unique structure that's only found in mammalian brains. So mammals have the ability with this limbic system to not only control memory, but it's also about learning. It's why we can learn complex tasks that other groups can't. But it's also very heavily involved in emotion and behavior. When you think limbic, the first word that needs to come to your mind is emotion. And then emotion directly affects the others. Think about memories of things. Those things that you have a very vivid memory of, they probably also have an emotional tie. You may have had a tragic death in your family, and the memory of that is tied to the emotion you felt with that. Learning, you may have really learned a lot from a particular teacher in school. Well, you're probably emotionally connected with that teacher as well. Your personalities got along. You felt comfortable in that room. Emotion has a lot to do with our behavior and our learning and our memory. And that's all the job of this limbic system of your cerebrum. So, 4 areas of the brain. I have the cerebrum, the diencephalon, the cerebellum, and the brainstem. So we've just covered the cerebrum, so we want to move now and talk about the diencephalon. Now location-wise, the diencephalon is literally at the center of your brain. Physically, if you go to find the diencephalon, it's at the center. The diencephalon is composed of 4 really important parts, and they each have their own nuclei, which just means gray matter. Remember that unmyelinated, well, my pen is not happy today. The unmyelinated, you get the idea, I can't write gray and matter apparently. That unmyelinated neurons that compose these regions in the deep, the thalamus, the hypothalamus, the epithalamus, the subthalamus. You can see the common root here. The diencephalon is all dealing with thalamus. This is where we process and integrate and relay information to other parts of the brain. It's where we maintain all of our homeostasis, blood pressure and body temperature, and go to sleep and wake up and be hungry and be thirsty. It's where we can regulate biological rhythms, your circadian rhythm, your ability to sleep and wake cycles, all controlled by this deep center of the brain referred to as the diencephalon. Now, the thalamus is a really important part of the diencephalon. The thalamus is your relay station. The thalamus is where all of your sensory input, except for smell, is sent. When vision comes in your eye, your thalamus says, oh, vision, occipital lobe, there you go. When a sound comes into your brain from your ear, it's the thalamus that says, sound, oh, okay, out to the temporal lobe you go. When all of our sensory input, with the exception of smell, comes into the brain, the thalamus directs it to its appropriate location. The thalamus is the ultimate traffic control center for the brain. Information is sent to the thalamus then routes it to the appropriate location of the cerebral cortex. That's why the bulk of the diencephalon, 80% of it, is the thalamus. The thalamus can also, interesting enough, act like a set of blinds. So think about blinds in a window. If the blinds are open, light comes in. If the blinds are closed, light doesn't come in. The thalamus can regulate what gets sent into the brain because it can function like a set of blinds. If the thalamus is open, the input will come in and continue to the cerebral cortex. But the thalamus can choose to shut out information as well. Interesting enough, the thalamus is highly influenced by the limbic system. So what did I tell you to think about with limbic? We want to think limbic, emotion. You can be very emotionally distraught, and your limbic system will tell your thalamus, shut my blinds. I don't want to learn anything. I don't want to pay attention to anything. I don't want to focus on anything. So the thalamus is the ultimate routing station to our brain. So to get information to the brain, you have to first get it into the thalamus. Hypothalamus is the big boss. He's the king of regulating all of your autonomic nervous system. Those things that happen automatically without us thinking about it. We sweat automatically. That's the job of the hypothalamus. Your female body releases an egg on time every month. Don't think about it. That is the hypothalamus. Your testes develop sperm without thinking about hypothalamus. We maintain not only body temperature but blood pressure. All of that, we are thirsty, we are hungry, and we don't get to think about when we're thirsty and we're hungry. It just happens. That is the autonomic nervous system being controlled by the hypothalamus. So if we look regionally for things, the thalamus is this big, huge bulk right here. Well, the hypothalamus is kind of like a funnel sitting on the end of it. So if we were considering this X is the room you're sitting in, if you look up, the ceiling is the side or bottom here of your thalamus. But the walls of the room, all the walls and the floor, that is your hypothalamus. It is linked by this little stalk to a very important gland called the pituitary gland. If the hypothalamus is the king of your autonomic nervous system, the pituitary gland is his head guard. The hypothalamus can send directions or commands to the pituitary gland, and then the pituitary gland will send those commands out to other organs of the body. The hypothalamus says, hey, pituitary gland, I'm thinking we need to release an egg. The pituitary gland says, hey, ovary, I need you to make and release an egg. The hypothalamus is that ultimate controlling center. It does so by sending out hormones. It sends out hormones that either tell things to happen, releasing, or tell things, no, wait, inhibiting. Then we have the epithalamus. So again, here is my big centrally located thalamus. Epi is to be upon, so here is the epithalamus. The epithalamus is caudal, which means towards the back. Maybe it's just blue, it doesn't want to write. All right, caudal. Whereas the pituitary gland out here in the front was rostral, towards my nose. So my epithalamus is superior to the thalamus. It's a lot of a gland called the pineal gland. So here is my epithalamus, and you can see the pineal gland there. That secretes the hormone melatonin. And melatonin is involved in your ability to go to sleep and wake up. It has a lot to do also with how you feel, your mood. The subthalamus is this region right up under it here. It is going to help control movement when it works together with a group called the basal nuclei. So these four parts make up my diencephalon. The biggest, bulkiest part is my thalamus. It's also my traffic control center. Then I have my hypothalamus, that is the ultimate king or boss of my autonomic nervous system. Then I have my epithalamus, that includes my pineal gland. And then I have my subthalamus region in here. That's going to help me control movement. So now we've covered two of our four parts. We've talked about our cerebrum here. We've now covered our diencephalon. So we lack our cerebellum and our brainstem. Cerebellum is posterior, or we would say caudal, and it's also inferior. Very, very, very involved with voluntary movement. Planning of voluntary movement, coordination of voluntary movement. Your cerebellum is sort of your part of your brain that lets you walk and chew gum at the same time. It's the part of your brain that lets you track the movement of a softball coming at you to know, okay, now's the time to swing the bat. It's the one that lets you sit at the stop sign, look left, look right, and see that car coming from the right and decide, Is it coming fast enough I need to wait, or do I might need to sit here, or go? The using of an instrument, the strumming of a guitar, the stringing of the violin, all of that is the job of the cerebellum. It, like the cerebrum, is divided into two hemispheres, but it's not quite so disconnected. Remember, I could take my thumbs and stick them into your longitudinal fissure and pull your cerebrum apart up to a point, the corpus callosum. I can't do that with the cerebellum. The cerebellum is actually connected to each other by this little bridge called the vermis. So if this was one cerebral hemisphere and this was the other cerebral hemisphere, they're not broken. They have a solid connection between them, and this connection is the vermis. Like I saw in the cerebellum is folded. The cerebrum had gyri and sulci. The cerebellum has ridges called folia. Their grooves, however, are still called a sulci or a sulcus. Now, why not just call them both gyri? Well, gyri are pretty big and broad, and folia are much more narrow and tight. But they do the same thing. They help increase my surface area. Also see, like the cerebrum, that the cerebellum has an outer layer of gray matter. We call it the cortex, just like we do in the cerebrum. So this is my cortex. But what is very different is that the bulk of the cerebellum is gray matter. And on the inside of each, let's see how well I can draw here. On the inside of each, though, is a big section of white matter that looks like a tree. So I'll draw it this way so maybe you can see a little better. That tree is called the arbor vitae because arbor means tree and vitae means white. Looks a lot like a tree. So look down here at a real section. Don't you see the big giant tree in here with its limbs reaching out? Most of the cerebellum is gray matter, that unmyelinated, slow, steady signals. When the central part, the white tree, the arbor vitae, is white matter, which is going to be myelinated axons that are traveling that rapid saltatory conduction. Then we have our brainstem. Your brainstem is the physical connection between the spinal cord and the brain. Vital to your survival because it controls a lot of just your basic homeostatic functions. Your ability for your heart rate to beat or your heart to beat and your breathing, all controlled by your brainstem. One reason why we worry about breaking C2, remember it has that dens on top of the body, and then you have your spinous process sticking out. If you break that dens and it were to pierce or sever your brainstem, you die because your heart and your lungs stop. Your brainstem also controls your reflexes, those things we don't have to think about, those automatic programmed responses that keep us from burning our hands or keeping our foot on a sharp nail. The brainstem has three parts: a midbrain, which is superior. In the middle is a pons, and then we have the medulla oblongata that connects it to the spinal cord. So here is my midbrain because that's my thalamus. Here is the pons, and then I have the medulla oblongata, which runs right into the spinal cord. The midbrain, sitting right up under the diencephalon, surrounds a portion called the cerebral aqueduct. It's simply a channel that's going to connect some of those internal ventricles to each other. This is going to get information in and out of your thalamus. It's going to carry motor fibers from the cerebral cortex and help talk back and forth to those basal nuclei. The pons is sitting right up under that. It has roles in regulating our breathing, and our reflexes, and your ability to fall asleep and wake up, our circadian rhythms. Then we have the medulla oblongata. It's the most inferior part of the brain. It does run right up into the spinal cord, so it's going to be right through that foramen magnum, that big hole in your skull. We do have tracts, bundles of myelinated axons that are involved in movement and sensation, and this is also regulating a lot of your homeostatic functions, back to your blood pressure and heart rate and breathing rates. In the brainstem is also a very important structure. Not as easy for identification, because when you look at a model, you can't really put your finger and go, "Oop, there's the reticular formation." You can pull a model and say, there's the midbrain, sitting right up under the thalamus, there's the pons, the bulge, there's the medulla oblongata running into the spinal cord. But for the reticular formation, the reason it's hard to distinguish is because it's gray matter. And it's gray matter in a big bulk collection. It's over 100 nuclei, bundles of gray matter, in the core of all three parts of the brainstem. So I want you to think about taking 100 pipe cleaners and bundling up in your hand, but then taking the top where they come out of your hand and spreading them out into like a web. That's really what your reticular formation is like. It's a very complex region of the brain that's getting information from the cerebral cortex, from your limbic system, emotion, memory, learning, and from all the sensory input, what we see, what we smell, what we taste, what we hear. And then it turns around and sends information back out to the entire brain and all of the spinal cord. The reticular formation is very involved in everything. Going to sleep and waking up, your arousal state, transmitting pain to parts of your brain, your ability to feel good or not feel good, your mood, and a lot of your homeostatic functions, blood pressure and breathing, and etc. So what makes the reticular formation difficult is it just can't put my fingers on it on a model. But what I want to remember, it's just a bundle of gray matter, nuclei, that can web out through all regions of the brain. So now that we've covered the parts of the brain, we want to talk about how do we protect the brain, because it is a vital organ to function. So we do put it in a protective shell. We have put it inside that bone casing, but we need to give it some additional shelter, because even though it fits fairly tight inside of that cranial cavity, there is a little bit of space. There's a little bit of space that would allow the brain to kind of flop around. So we're going to help shelter it through three things. We're going to use some connective tissue called meninges, and we're going to wrap them around the brain to help protect the brain. Now, we're protecting the brain from things in the body. We're also protecting the body from bacteria, viruses that might be in the brain. We're also going to use a cleaning fluid, a shower water fluid, referred to as cerebral spinal fluid. And it's going to do a little more than just shower the brain. Then we have a barrier, a blood-brain barrier, which we'll look at in a little more detail, but it is going to prevent things from coming into the brain that might possibly be in your blood, like viruses or bacteria. So we'll start with your cranial meninges. The meninges are what's going to help me fill up the space between the brain itself and the bone. But they're also going to serve a protective function. So if we start from superficial at the skull and move deep, then the order of what we should hit is first a space that's typically not there. It's called the epidural space, which is just a potential space. Typically, the meninges and the other spaces are going to fill up the area and push the brain up against the bone. So we don't particularly see an epidural space. You'll notice here in my list, there's not even one mentioned because it's only potential space. So the first thing we're going to hit then is a dura mater, which is actually two-layered. Then there is a subdural space. Then there is an arachnoid mater, a subarachnoid space, and a pia mater. So you can see these are the meninges, and these are the spaces found within them. So if I start with the epidural space, remember I said it is a potential space. Normally the dura is tightly bound to that bone, and there's only passage of blood vessels through this, so it's not really a space. The first thing we're going to encounter is the dura mater, which literally translates to tough mother or tough covering. It's the thickest of our meningeal layers. It's a lot of collagen with a little bit of elastic fibers, and it is double layered. The outer one is going to hug the bone. It's called periosteal, which literally means around the bone. The periosteal layer is going to be really, really tough, and it's going to almost anchor the brain itself to the frontal bone, parietal bone, occipital bone. The second layer in that touches the other meninges is called the meningeal dura. It is an inner layer, but it doesn't have any blood flow to it. It's mostly fused together with the periosteal layer. So in most brains, when you're dissecting them away, you can't really pull apart the two layers of the dura mater. They seem to be a single inelastic membrane, except in certain regions. So I'm going to give you kind of a big idea. So here is the cerebrum. So here is a hemisphere of the cerebrum, here's a hemisphere of the cerebrum. This is my longitudinal fissure. Remember I said I could put my fingers in your brain up to a point, which is the corpus callosum. And then here is your bone. So most often, your dura mater is simply going to run two layers inseparable up here, except when we get right here. This is where the periosteal layer will continue to hug the bone, but the meningeal layer will dip down in here into the longitudinal fissure. So this would create a region called a dural sinus, which is going to be like a drainage station for the brain. So if I'm working from bone in, we'll say bone, we don't typically have an epidural space. Then we have a two-layered dura mater. Then underneath it, I'm going to have a subdural space. This is filled with serous fluid, which if you remember from an earlier unit is a thin and watery fluid. This is going to house your veins, and this is where the veins are going to drain the blood flow away along with the sinus cavities of the brain. Underneath the subdural space, I find a very thin, irregular collagen layer referred to as the arachnoid mater. The arachnoid mater creates not only a thin, but a web-like membrane. And sitting right up underneath it is the subarachnoid space. This is where your cerebral spinal fluid is. This is also where a lot of your major blood vessels are. So I have bone first, a two-layered dura mater, then a subdural space, then a thin web-like arachnoid mater that gives me a subarachnoid space, and this is where my shower water is located. Unlike subdural, where I only have a thin layer of serous fluid. In here, I'm going to have my veins, but in here, I'm going to have a lot of your major blood vessels. Then the last layer is the pia mater. Now, the pia mater is the only layer that physically touches the brain. It is so in touch with the brain, you can't really separate them. If you had a brain in a dissection, to peel away the pia mater from the actual nerve tissue, nearly impossible. It follows every dip, nook, and cranny of the brain. Every gyrus, every sulcus, every gyrus, every sulcus. The pia mater flows along it. The pia mater is thin enough that it is permeable to fluids. It will allow material like glucose and oxygen to diffuse in and out through the cerebral spinal fluid into the nerve tissue. The next structural part we have are the ventricles. I said earlier that the brain is not solid. You have these cavities inside. These cavities are lined with one of our neuroglial cells, the ependymal cells. They're going to line it and assist in making the cerebral spinal fluid, or the shower water. So if I look at the chambers inside of the brain, I technically have a right and left. Let's see how I want to draw these. I have a right and left ventricle that's referred to as the lateral ventricles. You may think of them as ventricle number 1 and number 2. Inside of here, there are ependymal cells lining the entire ventricle. You get the idea, but you can see where I'm going with this. Ependymal cells lining the entire. They also have a blood supply referred to as the choroid plexus. So again, we would have the same design over here. See if I can do it faster. We have all the ependymal cells, and we have a choroid plexus. Now, they do resemble ram horns, although mine definitely does not look like ram horns, but they also have a horseshoe kind of shape to them. The two ventricles do have a connection in between them, a hole, that will allow it to connect to a more central third ventricle through this little hole. Let me draw my ventricles close together. This hole right here is called the interventricular foramen, and it leads to the third ventricle sitting down in here. So lateral ventricle, or 1 and 2. This is my third ventricle connected by my interventricular foramen. And then it has this little skinny channel referred to as the cerebral aqueduct that connects me to a triangular shaped fourth ventricle. Now, all of these are lined with epidermal cells. They also have choroid plexes, or these little blood capillaries within them as well. So I have my two ram horn right and left lateral ventricles, a hole in between my interventricular foramen, goes down to my third ventricle, then a long cerebral aqueduct connecting to my triangular fourth ventricle. This is going to allow me to make cerebral spinal fluid, drain it into here, with a choroid plexus make more cerebral spinal fluid and drain it into here, and with a choroid plexus make more cerebral spinal fluid and drain it. We actually make the shower water for the brain very deep within the brain in these ventricles. So here's a much better picture for you. But you can see the two ram horns. There's the lateral ventricles. There is my interventricular foramen. There is my third ventricle. This view is a little bit smaller, so I was drawing a little bit larger. Then there's my thin cerebral aqueduct, and because we're looking at both sides, you can see the triangle on each side. I only drew one side for you there with the triangle. And then that continues with what's called the central canal of the spinal cord. So you can see a little bit better here. There's my ram horns. There's my lateral ventricles. The interventricular foramen, my third. A little skinny cerebral aqueduct. See, now my fourth ventricle looks more like a triangle. And continues. These are the cavities deep within the brain that have cerebral spinal fluid inside. Now, this might look a little odd because normally if, I was a body and I needed to clean myself, I would need my shower water out here. Well, notice our shower water is actually deep within the brain. So we need to talk about how we make that shower water and how do we get it out, because to wash the brain, we're going to have to get it from the inside to the outside. The cerebral spinal fluid is clear. It's colorless. It's a lot like blood plasma, but it's going to protect the brain in several different ways. The brain does have a little bit of room in the cranial cavity. So I'm going to need to take that room, that subarachnoid space, and fill it with something. So cerebral spinal fluid is what we'll fill it with. It's going to help cushion the brain. It's also going to help keep the temperature of the brain stable. Way back in biology, we talked about all of the benefits of water. Water had a high heat of vaporization. Water is the universal solvent. Well, one of those characteristics is that water does take in a whole lot of heat before it ever elevates the temperature by a single degree. So because cerebral spinal fluid is very watery, it keeps the temperature of the brain constant in spite of elevations in your temperature because you go outside on a hot sunny day. So it cushions the brain. It helps it maintain a constant temperature. It also helps remove the waste from the brain. It's a way of getting waste filtered out. And then fourth, it increases the buoyancy. So think about what buoyancy is. It basically means it lets the brain float. Because you have a big cerebrum, you have a cerebellum, you have a brain stem, and then a little spinal cord. Well, remember, this is about 3.5 pounds. So I'm asking 3.5 pounds, which is fairly front heavy, by the way, to balance itself on this narrow brain stem and spinal cord. That weight alone would cause your brain to tip forward, which would either compress or pinch or actually break the brain off at the brainstem. So how do we prevent the brain from tipping forward and basically closing over on itself, even though it's in this little cranial cavity? We allow the brain to float. So we take the brain and we put cerebral spinal fluid in here, and it makes the brain buoyant. So it is floating in here, and it's not putting all that pressure on the brainstem and the spinal cord. How do we make it? We'll come back to those blood vessel little capillaries that I was drawing for you. They're called the choroid plexes. The choroid plexus is in each of the ventricles, and it's where the blood vessels come directly in contact with those ependymal cells. Now, some of the ependymal cells will make cerebral spinal fluid right by themselves, but they have to have a source for the water. So we need the choroid plexus. So if we come back to our ram horn right and left lateral ventricles, then we have our third ventricle, our cerebral aqueduct, our fourth ventricle. We'll connect these with an interventricular foramen. The choroid plexus found in each of these is what I'm going to use to filter out the water. We're going to get about 2/3 a cup of it circulating through the brain and the spinal cord. Now, we make it all the time. We're constantly making it because that's why I wanted you to think shower water. You can't think bath because bath is something you sit in, although it's comforting, you're soaking in dirty water. Shower water is good because it comes fresh out of the faucet and drains away. Fresh out of the shower head and drains away. Fresh out of the shower head and drains away. So we are constantly making cerebral spinal fluid, I'll color it black, sending it through these ventricles, making cerebral spinal fluid. It's escaping out of these little doors called apertures, and then it is surrounding the brain, and it will rinse out through the dural sinuses. The sinuses are like the drainage stations. So continually, we are making cerebral spinal fluid through the 4 ventricles. We're letting it out the apertures. It is going around the brain, rinsing the brain, keeping the brain floating, and it's being completely replaced every 5 or 6 hours so that we're constantly having clean shower water wash the brain. So here's an excellent look at it. Here's one of my lateral ventricles. There's my third ventricle. There's my cerebral aqueduct, my triangular fourth ventricle, and you can see my little choroid plexes. So what we're going to do is we're going to filter fluid out. We're going to filter fluid and electrolytes out of the capillaries of these choroid plexes. The ependymal cells will take them then and secrete cerebral spinal fluid into the ventricles. We will secrete cerebral spinal fluid into the ventricles. We will secrete cerebral spinal fluid into the ventricles. And then there's that escape door I was telling you about. They're called apertures. We will let it then escape and go out and completely wrap around the brain until we get to our drainage stations, which are our sinuses. So you can see the sinus here. This is our exit route. So we'll drain out. It's just kind of like a backward shower. The water's coming from inside, and then it goes until it drains out. Every 5 or 6 hours, we're completely turning over our cerebral spinal fluid. But in the time that we have it, we are letting the brain be rinsed and fed, and we are cleaning and letting the brain be buoyant. I shouldn't have said fed. I meant rinsed. We take away waste, and we keep it buoyant. To feed the brain, you need the blood-brain barrier. Now, the blood-brain barrier is not an actual physical barrier. It is a characteristic of the capillaries that feed the brain. So notice, here is a blood capillary. You can see it's made of a very thin cell, and it's kind of leaky. See these gaps in between them? Well, right now, whatever bacteria or solute or nutrition is just easily going out of all these little cracks. No problem. Well, it is a problem when you get to the brain because we do not want the brain to just have free access to what's in the bloodstream. So the blood-brain barrier is a series of 2 structures. It is the astrocyte, which is one of our microglia cells, and our zipper tight junctions. So what we do is instead of allowing free leakage like you see in this image, we use the feet of the astrocyte to cover up some of these sections, and we zipper them with tight junctions. So by covering up and zipping together these cracks, we ensure that anything that gets out of the capillary must physically go through the capillary's cell wall. What it's going to do is help us limit disease-causing organisms from leaving the bloodstream and entering the brain. But it does allow things that are lipid-soluble, like glucose and our oxygen and some of our nutrition easily gets out through these astrocytes and tight junctions in time to feed the brain. So you can see what easily goes through: water, oxygen, carbon dioxide, anything that's not polar and it's lipid-based. There are some proteins along the way that can help me pump other things through, like glucose and amino acid and sodium and potassium, things that you need to get to your brain. What cannot get through? Large polar molecules. We basically say, nope, you are not coming through to the brain. Now that's good because viruses and bacteria, they don't get to the brain. But it's bad if you have a brain disease that we need to deliver medication. We can't deliver it through the bloodstream because the blood-brain barrier, this capillary property, is actually going to block the medication. Now, what I don't want you to think is that because we say barrier, that it is a physical wall, because it's not a physical wall in your brain. Instead, it is the property of all the capillaries in your brain. Each capillary has a barrier that prevents stuff from leaving the capillary and just getting free access into your brain.